So, welcome to Bio 260. We're going to be talking about biostatistics throughout the semester. Let me actually stop my videos because I feel like that could get a little distracting. Okay. So just basic contact information. So my name is Kira McFarlane. I'm a master's student here at Cal State Long Beach. I'll talk a little bit more about that in a minute. So my email address is, Kira McFarlane at student.csulb.edu. That's on the beach board as well. So all of my contact information is there. When you email me, if you email me, please include Bio 260 and your section number in the subject line so that I know what you're talking about. And then of course, any relevant information, like lab 2 question or homework issue or R problem, something like that. And then my office hours are Wednesdays and Fridays from 12 to 1. Those can be flexible if that doesn't work for a lot of people. I may even do like a little, I don't know, questionnaire or doodle poll or something to figure out when a good time works for everybody. Yeah, but that's what they are for now. So again, for this class, you have to have the lab manual, kind of, not necessarily the newest copy, but try. And please read the scheduled chapters before you come to class. I know a lot of people are still getting the lab manual, but once you do, please try to read the chapters beforehand. It'll make everything run a lot more smoothly. This class is mandatory, so you need to be here. I'll be looking at the Zoom recording logs at the end of every class to see who stayed the entire time. As I'll show in I think another slide or two, that you get participation points for being in the lab, at least until I say that you have permission to go. There's a lot of times where the lab won't take the entire 2 hours and 45 minutes. In those cases, I'll probably let you go early as long as you finished the lab material. If for some reason you can't attend a lab, please just let me know. Send me an email. You don't have to go into detail about whatever's going on. Just, hey, something's come up. I can't come to class. And then we'll try to work out a different, lab for you to attend. If that's not possible, then I will be posting all of my lectures on YouTube. So yeah, homework for classes you did not attend will not be accepted. So the grade breakdown for the class is we've got participation, which is 5 points per class, per synchronous class. So every class that we actually attend together. There's a couple of weeks where there's, like Veterans Day or something like that, that would cancel a Monday class. On those classes, or those weeks, we won't have a synchronous class where we all meet at the same time. You'll just have an assignment that you need to do at home. So no participation points for those. There will be 8 homework assignments worth 10 points each, 1 quiz that'll take 45 minutes for 35 points, a group project, which will be 35 points, which we'll discuss closer to when that time comes, and 2 lab exams, each worth 100 points. And those will take the entire lab period for a total of 400 points. So some of the breakdowns, timing, number of assignments, things like that might change throughout the semester, but this is what we're working with for now. So yeah, hopefully everybody's checked out the syllabus. Sorry it took me a little while to post, but it's up there now. So the lab midterm, week of October 12th, final will be a week of December 7th. Quiz 1 will be the week of September 28th, and the group project is due the week of November 30th. Again, those might change, and we'll discuss particularly the group project more when the time comes. So the reason that I had everybody do the pre-lab survey, which everyone has hopefully already done, but yeah, so the reason that I had everybody do that is so that I can put you into pre-assigned breakout rooms based on your interests, your majors, what you want to do after graduating, things like that. We're really trying to build like a sense of community with everything, because I know some people are feeling a little bit isolated being just online all the time, and not really interacting with other students. So hopefully having a group that you're with through the entire semester will help with that a little bit. And it makes it a little bit easier to ask questions and get help on anything you might need help with. But yeah, so please do the pre-lab survey as soon as possible. If you haven't, I'm offering 5 points extra credit if you complete it before 5 p.m. on Friday. And then, of course, cheating and plagiarism will not be tolerated. Please don't do it. It doesn't benefit you. It might get you through this class, but in the long run, it's not worth it, really. And if I suspect you of cheating or plagiarism, you will automatically fail this class and be reported to the college. So, okay, so a short little icebreaker. So I'm going to actually put you into, well, okay, sorry. First, are there any questions? Does anyone have any questions about any of that stuff? Okay, cool. Also, I'm gonna ask that when you speak, you turn your camera on. I know that like everybody is in different situations and might have different stuff going on in their backgrounds, but it gets really sad just staring at people's like screens and just names constantly. So if you could please turn on your camera when you speak at the very least, that'd be great. But yeah, so I'm gonna actually put you into breakout rooms for like 5 minutes just to get kind of acquainted with the other students in the class. Hopefully you all have my PowerPoint downloaded. There's like a list of like 30 random questions that you could ask, but feel free to honestly like, you know, talk about whatever. Just like to get to know each other a little bit. So I'll do that for about 5 minutes and then we'll get together. I'll introduce myself and then we'll finally begin the lab. Okay. So a little bit of background about me. Let's see. So my name's Kira McFarland, right? So I'm a master's student here at Cal State Long Beach. But I actually got my undergrad degree, my bachelor's degree at UC Santa Cruz. We are the banana slugs, which is why I included this picture of banana slug here. They're really cool. We get like to be like 10 inches long and they're everywhere in the forests. And I don't know, slugs used to really gross me out, but now I think they're kind of cute. So yeah. When I was there, I did my degree in marine biology with a focus on cetaceans for the most part. So after graduating, I moved to Washington State for a while, and I did an internship at a place called Cascadia Research Collective, where I was mostly doing some whale fluke IDing for humpback, gray, and blue whales. But sometimes I got to go out on whale watching tours and photograph the flukes myself. I did a couple of necropsies on like porpoises and things like that. It was a really, really cool experience. After that, I moved to Canada for about a year, where I worked with this group called CETUS, Research and Conservation Society. And we'd kind of like zip around on this little Zodiac out in the Salish Sea and pretty much prevent and document any incidences between the southern resident killer whales and boaters, including the whale watching tours that I had previously been on for my other job. So that was really cool. But now I work with mice. So now I'm in the wetland ecology lab with Dr. Christine Whitcraft, and I catch mice in a tidal wetland along an elevation gradient from lower marsh to higher marsh. And the lower marsh, when tides are high, the vegetation is completely covered compared to high marsh where it's only covered sometimes. So I'm looking at a couple different species and seeing how they're distributed across the marsh area. And that's me holding a pretty upset looking mouse. But yeah, so that's me in a nutshell. And with that, we will just begin with the lecture. So chapter 1 is summation notation. So summation notation uses the capital Greek letter sigma, and it pretty much denotes to sum variates in a data set. So, but first, let's get some terms out of the way. So a variable is any attribute of interest recorded during research. It's a variable, a characteristic, yeah. An index tells you which value in a list to refer to, and variates are the actual data values. So in this example here, we've got a one-dimensional data set. So we have two columns, but the first column is the index. So we've got from 1 to 10, and then we've got the weight, the variable is weight from 1 to 10, right? Those are different actual values for weight. So the variable is weight. If we saw something written as x sub 1 equals 34, we know for weight at index 1, the variate equals 34, right? For weight, which is our variable x, at index 4, our variate equals 37. When we use sigma, the capital Greek letter sigma, we're saying that we want to add up a series of numbers according to the index. So if we wanted to add all of the numbers from this list, we could write it out as x sub 1 through 10, all summed together equals 331. Or we could just write sigma from index 1 to 10 of weight equals 331. Yeah, so this says sum all indices of variable x from 1 to 10. You can also only include some of the variates instead of all of them. So in the problem below, it only wants us to include the observations 1 through 8. So we're summing all indices from 1 to 8 of x, of weight. So again, that would be the equivalent of writing it out like this. And equals 266. In this case, the notation is telling us to sum from index 3 to 6. Right, so from 3 to 6. Let's see. Are there any questions there? Let's see. I don't think I can share that and have the chat open at the same time. Fortunately, don't think so. Okay, so I'll just check in a little bit. Okay, so we can also perform transformations on the data before summing up the variates. So, and we have to remember to pay attention to the order of operations when we do this, right? PEMDAS, right? Parentheses, exponents, multiplication, division, addition, subtraction. So with the way that this is written, we want to square each variate first and then sum the variates from 1 to 10. So each individual variate from this data set is squared and then added together. And we get 11,073. In this example, we're actually squaring the sum, the total sum of the variates. So the sum of the variates from 1 to 10 is 331. That is the value we want to square. And we get 109,561, right? So those are very different numbers, right, between these two slides. So it's super important to make sure you pay attention to the order of operations when doing these problems. Okay, so these examples are on page 3, but I wrote them here as well. So if you don't, if you're wondering why I'm wearing different clothes, it's because the first half of lecture on Monday recorded, but the second half did not. So I'll be going over the in-class work-through, the Excel workbook, all of that now again, and then going into the second half of the class and the rest of the Excel workbook to cover everything. Okay, so starting here from the Excel workbook, on the first page, we've got this function, new functions, we've got chapter 2 practice, so on and so forth. We're going to move through each of these tabs. So right here, we've got the new functions that we're using for this lab. We've got sum, which sums the chosen cells together, average, which finds the mean of the selected cells, the minimum finds the minimum, max finds the maximum. So before we really get into using and doing and solving these summations, let's first go through some of these functions. So when we look at what we have here, we have a series from 5 to 9. If we wanted to find the sum of this in Excel, we could type equals sum and then select the cells that we're interested in, close the parenthesis and hit enter. You could also do sum and type the cells. So let's say this is A, excuse me, A4, so I could type A4 colon A8, close the parenthesis and hit enter. But it's a little bit more likely that you're going to make an error doing that, so I like to just select the cells. At number 2, we see that we can also do basic math using the plus, minus, slash for division, the asterisk for multiplication, and the caret for exponents. We could do more complex equations if we include parentheses to separate the equations. But right here, we're just trying to find the sum of these two cells, of 4 and 5. So we can do this one of two ways. We can type 4 plus 5, and we get 9, of course. We could also do equals this cell plus this cell. Excuse me. And we also get 9. So the nice thing about doing it that way when we refer to specific cells is that coming down here for number 3, that means that we can actually drag and drop. So if we scroll our mouse over this little box at the bottom right corner, we get this black crosshairs. If I click that and drag downward, we see that the equation here was actually copied into each of those cells. So looking up here, you can see that it changes for each of those cells. And Excel does this by a kind of distance association, right? So it knows, okay, you're typing in C24 and you're adding A24 and B24 to find the sum in C24. So here, Excel's like, oh, you're adding A25 and B25 to get C25. So by scrolling downward, it knows that we're trying to add these two adjacent cells to get the sum in the third cell. So that's how it's doing it. It's keeping these spatial relationships the same. For number 4, we see that we can use this $ to anchor cells when we use this drag and drop. So up here in the equation line, I have A $33 plus B 33. So A33 is this cell, B33 is this cell. But by putting the $ before 33, I'm anchoring number 33. So when I drag now, so all of these numbers are the same as the numbers up here, but we see that we actually get different numbers below now, right? And that's because looking at that equation, we see that A33 has remained constant. By using the $, we've anchored cell A33, but allowed the second component, this B34, well, it was B33 in the previous one, right? And that is changing. So Excel is saying, oh, you want to keep A33 the same, but with dragging them, you want to change the other one in relationship to the cell you're putting the equation in. Right, so you can see that up here, that A33 stays constant, but the B column changes downward. The next one, we have sequencing. So dragging can be used to copy numbers and sequence numbers as well. So here we've got this kind of jumble of letters, and then we've got 1 and 2. If I click 1 and I drag just only the 1 highlighted all the way down, we see that Excel just copies the number 1 all the way. Same thing with 2. If I click 2 and then drag downward, I just get the number 2 copied over and over. But if I highlight 1 and 2 together and then drag, Excel realizes that we're making a sequence of numbers. And so we actually get a sequence from 1 to 7. Sorting is also another useful function. So again, we've got this kind of like jumble of letters, but this nice sequence from 1 to 7. If I select all of these letters and then come up to the top right corner here and click sort and filter and sort from A to Z, I get this warning. So it says, do you want to expand the selection or continue with the current selection? I'm going to expand the selection first. So now the letters are in alphabetical order, but the numbers next to it are no longer in order, right? So let's look at that again. We have 1, or A and 1, D and 2, G and 3, right? So these are associated with each other across rows. So when I sort and I expand the selection, Excel expands the selection into the associated rows, right? So A is still with 1, B with 7, C with 6, so on and so forth. So it shifted the entire row based on the column that I selected. And so I just undid that. So we're going back now, we're selecting the mixed up letters again, clicking sort and filter, sorting A to Z. But this time I'm going to click continue with the current selection. And now we see that it only sorted the selected column. It didn't expand into the second column. Last is the data analysis tool pack, which we're not going to use in this class today, but actually later on in the semester, this is going to be really useful in the first half of the class at least. The data analysis tool pack allows you to do all sorts of analyses. So you want to come up here into the ribbon, that's this big gray area. And this is the instructions I'm about to show you are a little bit different between Mac and PC. There are instructions on Beachboard under the course materials for the R and data analysis tool pack installation. And there are instructions for Mac there, but they're pretty much the same. Anyway, so you want to come up into this ribbon, this gray band up at the top, right click to customize the ribbon. When you get this box, you click on add-ins. It takes a second to load. And then you click on this analysis tool pack. Now, it might seem like you want to press OK. You don't want to press OK yet. You want to click on Go. And then check this box. If you don't have the data analysis tool pack, it'll be unchecked. So go ahead and check it, then press OK. Now, if you come up at the top to data, on the far right side, you should have this data analysis. And when you click this, this window full of different statistical analyses comes up. And most of these probably don't mean anything to you right now, but they will by the end of the semester. Okay, so from there, let's go on and do some of these practices for summations. So I've done the first one. So looking at this, we are looking for the sum from rows 1 through 5 in column 1, right? J has been replaced by the number 1, which means that we're talking specifically about column 1. We're keeping the column constant, but the row varies from index 1 to 5. So that's what I've done here. Column 1, rows 1 through 5. For the next one, we're looking at columns 1 through 3, J through 3, J equals 1 through 3, I equals 1 to 5, so rows 1 through 5 for values of X. So that actually means the entire data set, right? Columns 1 through 3, rows 1 through 5. That's the entire thing. So we're going to type equals sum and then select the entire data set, close parenthesis, enter. For the next one, we have the sum from index I equals 1 to 5, so rows 1 through 5. J, there is no J, we have the number 2, which means we're talking about column 2. So column 2, rows 1 through 5, that's what this area is being referred to. So the sum of column 2, rows 1 through 5. Close parenthesis, enter. Here we've got, so you'll notice that I and J are switched. It doesn't really matter though. So we're still talking about rows 1 through 5 because I equals 1 to 5, J equals 1 to 2, so columns 1 and 2. So rows 1 through 5, columns 1 and 2. So sum, columns 1 and 2, rows 1 through 5. This one, there's a little bit of a typo. This I, both of these I's should be J's. So that would be column 1 through 3, row 1, because I, what should be I, is being kept constant at 1. So we're talking about row 1, columns 1 through 3. For number 6, we've got a word problem and the notations are blank. So on your own, if you're doing this again, I'd go ahead and pause the video and actually write them down. I'll show you those as well in a second. Yeah, so find the sum of the second and third columns. Because rows aren't specified in this, I'm assuming that it's all rows. So the sum of the second and third column, all rows. And I think I can actually just draw straight onto Excel. So the sum of the second and third columns would be the sum, we'll say j equals 2 through 3, and then the sum I equals 1 to 5, so all rows of x sub I j. That's what that one is. For number 7, we have the sum of the first and second rows, all columns, assuming because it's not specified. So the sum of the first and second row. Written in summation notation, that would be the sum of all columns, right? So 1 to 3, and the sum of only the first to second rows. What is the sum of all these squared values? So this one's a little bit different. Let me move this to a place that makes more sense. So what is the sum of all the squared values? You have to be careful with order of operations here. So there's actually 2 ways to go about solving this one in Excel. They do the same thing. So we've got the sum of all the squared values. That means we want to square each value first before we sum them together. So one way to do that is to do the sum, then open a parenthesis, select all of the values, close the parenthesis, caret 2. So we're squaring, that's telling Excel that we want to square each of those values and then sum it, right? So we're working from the inside out. In the inside, we have each value, we're squaring that. Outside of that, we're summing them. Another way to solve that is to actually recreate another data set, right? So let's say I'll click right there, hit equals this cell. I'm going to square that cell. And now I have the square of 58 right here. If I click that and drag it over, I have the squared values of the cells above it, right? Because Excel works on distance and spatial associations. If I highlight all of those and drag down from there, I get all of the values squared. This row is zeros because I went 1 too far, so it's squaring this row here, which is empty. So I'm just going to delete that. So now I have a second 2-dimensional data set with all of the squared values. So another way I could solve this is now to just sum all of those together, right? So sum of all of these values. And we get the same number. What is the squared sum of all the values? So the way this is worded is that we're going to first sum all of the values and then square them. So find the sum of all of the values and then square that. sorry, and I forgot to write number 8. So number 8 would be the sum from j equals 1 to 3, and the sum of I equals 1 to 5 of each value squared. For number 9, we're summing all the values and then squaring it. So number 9 would be each value, so from column 1 through 3, row 1 through 5 of each value, but the sum is what's being squared. So parentheses go all around that, and we're squaring that. For number 10, so we're finding the sum of all the squared values in rows 2 through 5 in columns 2 and 3. So the sum of the squared values, so that's using this again, right? We're squaring the values before we're summing them. So sum, open parentheses, I'm going to do it both ways again. We're looking at rows 2 through 5, but only columns 2 and 3. So this area of the data set. Closing the parenthesis, squaring it, and then closing the other parentheses. Using this set over here, I'm going to do sum of, rows 2 through 5, columns 2 and 3, so here. And again, we get the same number. Written in summation notation, that would look like this. So I'll keep it consistent with doing J first. So J, so columns 2 through 3, rows 2 through 5, with each variance squared first. So the square goes with the x instead of around the entire thing. Okay. So that's summation notation. So next we'll go on to accuracy and precision. So let's talk about variables. So what is a variable? A variable is just the observation or observations or a phenomena being observed. It could also be called like a characteristic that you're interested in recording. And there are different types of variables. There are continuous variables, discrete, ranked, and attribute. Sometimes those are more broadly described as just like categorical and numerical, or nominal and numerical. There's some other terms for them, but these are the 4 that we're going to discuss. Attribute is often called categorical or nominal, though. I'll usually refer to it as categorical. And I'll talk about that more. There are also different statistical analyses. So we have descriptive statistics, when we collect data from a population and describe the characteristics, right? We're describing, so we're using descriptive statistics. And these are often, like for my study, for example, mice sizes. So I take tail and foot and weight measurements, sex, species, and habitat type. Those are describing the population that I'm studying. There are also inferential statistics, which are used to answer one or more questions or hypotheses, and it's a more experimental approach. Chloe Van Groothuist is another student in our lab, or I guess she actually just graduated, but her study was looking at the metabolic rates of gobies. So she hypothesized the gobies utilizing altered habitats will have a higher temperature sensitivity than gobies using natural habitats. Going back to the variables, we've got continuous variables. So these variables can theoretically take on an infinite value, right? So height, weight, pH, temperature. There are theoretically a million trillion infinite number between 1 and 2 pounds, right? It's just that realistically we can't measure to 100 points after 0, or points after the decimal. So we have to be realistic in our measurements and our expectations for them. So that brings us to the implied interval, or the implied limit. So the difference between 2 millimeters and 2.0 millimeters, right? If I say that something is 2 millimeters, we're working with a scale of millimeters, right? It may actually be 2.4 millimeters, but I probably rounded down to 2 millimeters. Whereas if I say something is 2.0 millimeters, there's a higher implied accuracy, right? Now we know that we're working with a greater accuracy. 2.0 millimeters truly means that it's actually anywhere from 1.96 to 2.04 millimeters. In both of those cases, we would round to 2.0. Most of you are probably already pretty familiar with significant figures, so I won't really go over it too much. If a number is greater than 1, all figures are significant figures. If a number is less than 1, figures after 0 are included in significant figure counts. For assignments and quizzes and exams and things like that, I'm going to ask that you round to 3 points after the decimal, so 3 digits after the decimal. Continuing on with the variables, we've got discrete. So discrete variables are whole numbers, they're integers, right? You can't have 3.5 people, you have 1, 2, or 3 people. In some studies, though, where you're looking at count data, a lot of the times you'll still encounter non-integer numbers, and that's because you're actually looking at proportions. So in this example right here, we're looking at a count of 10 people, 7 are female, 3 are male, which means we have a proportion of 0.7 females and 0.3 males. Next, we have ranked variables, where there is a significant order of the variance. So the order of the variant is meaningful, but the magnitude is not. Example of this is finishers in a race, right? So first, second, and third place may be separated by, 10 seconds to a minute or something like that, but that's not really what counts or what is being measured, right? It's first, second, and third. It's the order that they're coming in. And lastly, there are attribute / nominal / categorical variables. And these are essentially non-number variables. So names, classes, categories. I have color, age, and sex. Age is a number, but a lot of the time age class is used, right? So we might have, juvenile, adult, senior. Those would be categorical, attribute, nominal variables. Okay, so accuracy. Accuracy is actually referring to a specific thing, right? We tend to use accuracy and precision interchangeably in just like everyday language, but scientifically there's actually, they mean different things. So accuracy is how close the observed value is to the actual value. But you have to be practical though. So when I'm doing my studies, right, I'm handling live mice, and they're wiggling and they're trying to bite you and they're trying to escape. So I have to be realistic with my expectations of how accurate my numbers are when I'm trying to get measurements. And it's not realistic for me to take like a caliper out into the field and try to do it that way. Precision actually refers to how close the repeated measurements of a single object are to one another. So it's a measurement of the variation, the variability in the measurements that you take. So something can be precise but inaccurate or vice versa. And this is a good figure to kind of show that. So here we've got high precision. All of the points are close to each other, but low accuracy. We're actually pretty far from the true measurement or the bullseye. Here we've got high accuracy. It's pretty close to the center, but the precision is low. The points aren't very close to each other. There's a lot of variation, a lot of dispersion in the data. Here, the points aren't precise or accurate, right? They're not on the center and they're not close to each other. Whereas here we've got both high precision and high accuracy. And lastly, there's the stopping rule. So this one's a little bit harder to understand and not quite as intuitive, but it's actually very important for having comparable measurements across a study for different trials. So one example is a pattern of turtles swimming by a certain point measured in time. So how many turtles swim through a culvert in 1 minute? The 1 minute is the stopping rule in this case. You don't take measurements before or after. It doesn't matter how many turtles swam by before the minute started or after the minute started. The 1 minute time interval is the data set, the sampling unit, right? And it's important to have that sampling unit so that you can compare sampling units consistently through multiple trials or multiple years. Okay, and that's the whole PowerPoint. Let me switch back to the Excel. Okay, so some of the sample statistics that we're going to calculate are mean, minimum, maximum, and range. So mean refers to all of the numbers in your data set added together, summed together, and then divided by the sample size, the number of variables in the data set. When we calculate the mean in Excel, we don't do so by typing mean, we actually type average. The reason though that we don't use the word average often in science, and we typically use the word mean, is because average sometimes actually refers to medians, the middle value in a data set. So it's more accurate to use the word mean. So we're going to calculate the average though, because it's the term for Excel. And we're going to select the data set, close the parenthesis, and hit enter. And we get 7. The average or the mean for this sample is 7. We can then calculate the minimum, typing min, selecting the data, closing the parentheses, hitting enter. We find that the minimum for this data set is 5. Same thing for the maximum. Close the parenthesis. The maximum is 9. And the range is just the maximum value minus the minimum value. So I'm going to select the maximum minus the minimum. And we find that the range for the data set is 4. Okay, and then last activity. If we were in class, we would have given you things to measure using a ruler and a caliper. Let me look up really quick what a caliper is, because I don't think a lot of people know what it is. Caliper. So this is a caliper, and it's just a more precise tool for measurement, right? So you can see it actually goes to like the thousandth space. And in the in-class experiment, you would have found that calipers are more accurate and more precise than the ruler. It's a pretty straightforward experiment, but so we just provided you with the data for that. But calculating the mean for each of these columns, the minimum, and the maximum, and then finally the range. So, right, the maximum minus the minimum. And then using that drag and drop that we learned earlier, we're going to actually just pull that all the way to the end here. And now we have those 4 sample statistics for each of these columns. And then I'm going to copy these here. So if you just copy and then paste, you'll see you get this value error. And that's actually because it's copying this cell association like I was showing before, but it's trying to subtract this blank cell from, or this word from this blank cell, which is not possible. So what you can do instead is copy and then paste as a value. And we'll do that for each of these. Oops. And just looking at this, we can see that the calipers are more accurate and more precise than using the rulers. And we can actually visualize that data very easily. Yeah, and we see, looking at this, that we have a much higher range for the rulers compared to the calipers.